Dear potential future students of the University of Pannonia, welcome to this video. My name is Mihály Pósfai. I'm a professor at this university, and I'd like to explain some of the research opportunities that you could find at our university. And I hope that by the end of this talk you will be convinced that it's worth coming here and it's worth to pursue some sort of scientific research. So where is this university at all? So we are located in the middle of Europe. This is Hungary, and this is the city of Veszprém. We zoom in and you can see that Veszprém is just about 1.5 hour drive from the capital, Budapest. And we are next to this beautiful big lake, Lake Balaton. And Veszprém is a city of about 60,000 people. It's a nice, quiet city. Students love it who come from huge mega cities because it's really a good place for doing studies and research. And since I'm at the Faculty of Engineering, I'm supposed to talk about research groups or research that we are doing at this faculty. Well, our university has a very strong research performance in some disciplines that are listed here, and many others that I'm not going to talk about. But of course, in a 15-minute video, it's impossible to talk about all research groups and all the research lines that we are busy with. So I'm just going to talk about my own research group, and even within that, only about one certain research direction. We are based in this so-called Nemo Lab. You see this building here, which is an electron microscopy center. On the right hand side you see this is the electron microscope that we are using in our research, and we use it for looking at nanometer scale objects, mostly minerals, because my group is called an environmental mineralogy research group, and we have very diverse topics. For example, we look at sediment formation in lakes. Of course, this study is motivated by the proximity of Lake Balaton, and we are very fond of this lake because Hungary doesn't have a sea, so it's very important both economically and culturally in Hungary. And we are interested in the ecology of the lake and how minerals, the sediments, what role they play in the life of this lake. Sometimes there are some blooms of algae, and sediment minerals have a certain role in this, and we want to understand that. But I'm not going to talk about that. This was just a teaser, a trailer of that. The other topic we are doing is biomineralization. This is a very interdisciplinary field because, as you might know, actually I don't know what is your background, so probably you are watching it. Many of you have very diverse backgrounds. So I decided to talk about these magnetotactic bacteria and biomineralization because that's a very interdisciplinary topic. So there is some physics in it, there is some earth sciences, biology, chemistry, nanotechnology. So I hope that each one of you who is watching will find something interesting in it. So biomineralization is a process by which minerals, inorganic crystalline compounds form because of by living organisms. Well, one example is our teeth and our bones. Those are also biominerals. But there are some simpler organisms that are also doing it, and among them magnetotactic bacteria are the first. So what are magnetotactic bacteria? It's relatively simple to have a view of them. So here is a recipe how to do it. You go out in the field to a pond and collect some mud, either from under the ice or just in a simpler way. You take this mud in the lab and put a magnet, just a bar magnet, next to the sediment-water interface and wait about 30 minutes. And then you take a droplet from here, just from next to the magnet. You take a droplet with a little bit of sediment and water and put the droplet under the optical microscope, and again put the magnet next to the droplet. And you will see something very interesting. You will see that all these organisms, there are some sort of organisms, that rush to the edge of the droplet. And if you turn the magnet, then they all race away. Now again, turn the magnet, they come back. So this is how, actually you can play around with these poor bacteria. They follow the magnetic field. So the bar magnet has a stronger magnetic field than the Earth magnetic field. So all these bacteria think that this is the way to north, and they come down. If I turn the magnet, they think that way is north, and then they will leave. Of course, this is a very strange behavior. Actually, it was an American professor, Richard Blakemore, who first noticed this behavior of bacteria in 1975, and he was very surprised. And of course, the question immediately arises, how do they do it, and why do they do it? So for the question of how they do it, it's very simple. Here is a cell of a bacterium, a magnetotactic bacterium. It's just a few micrometer large, and it almost looks like it has a backbone. This backbone is made of magnetic nanocrystals of the mineral called magnetite. Here's the chemical formula Fe3O4, so it has iron and oxygen in it. It's an iron oxide, and because of these magnetic nanocrystals, the whole cell becomes like a compass needle, and it gets tilted, rotated along the Earth's magnetic field, and then it has to swim along the field lines. And the next question is, why do they do it? Well, I told you that they were swimming towards the north, but that happens only in the northern hemisphere. In the southern hemisphere, they swim towards the south. But actually, they are not interested in going to north, but they are interested in going downwards, because they live in places where there is water and sediment, and there is a gradient in oxygen concentration. There is a lot of oxygen here, there is none here, and there is this layer where the oxygen just drops to zero, and this is where the bacteria want to live. This is their habitat, this very thin layer. And if you are a bacterium and you are not magnetotactic, then you have to search for this layer, and you go do a random walk. You just go this way, that way, and you sense the oxygen, and if it's okay, you stop here. However, if you are magnetotactic, you are forced to swim along the field lines. And these field lines not only point to north, but also deep down. If there is a storm, stirs up the sediment, or these magnetotactic bacteria get out of their preferred habitat, once it's calmed down, they immediately find it. So actually they are very efficient finding their optimal habitat, because they reduce a three-dimensional search problem into just one dimension, because they have to swim along these field lines. So this is an evolutionary trait of these bacteria. At least this is what we know about them at the moment, but there are still many things to be discovered. So why are we interested in them as environmental mineralogists? Well, first of all, because I'm interested in these magnetic nanocrystals. So these bacteria have many, many different types, and each bacterial strain produces magnetic nanocrystals of specific shapes. That's very, very interesting. Actually, some bacteria produce these octahedra. That's not so surprising. Magnetite is a common mineral in rocks, for example in basalt. And if you look at magnetite from basalt, you will see that magnetite is always octahedral. However, these have some more complicated shapes, and this is very strange, almost looks like a barrel. And all of the crystals have these shapes. And these are really weird. Actually, in the laboratory, we cannot produce such sort of magnetite crystals. And of course, we are very interested in knowing how the bacterium does it, because magnetic nanocrystals have a wide range of applications, and we would like to know how to make these elongated crystals, for example. So, first thing is to characterize these nanocrystals, and that's what we are doing with the electron microscope. And then the next step is how we can use the information that we got from the bacteria for making these nanostructures. So I'm just showing a few examples, like we study the crystal structure of these magnetosomes. They are called magnetosomes. And here you see almost atomic resolution image. You see this is only 5 nanometers, and this magnetosome has a defect here. It's a twinned crystal. So with the electron microscope you can go down in resolution to the atomic level. But you can also study the chemical composition with the electron microscope. Here's an example. You see the iron distribution. These are the magnetosome crystals inside the bacterium, and the green is the phosphorus. And you see a rim around these particles, and that's because these particles grow in a phospholipid membrane. It's a biological membrane that surrounds them, and it has a lot of phosphorus, and you can see the phosphorus distribution here, that it's all around these crystals. Physicists are very interested in nanoscale magnetism, and these bacteria provide a natural laboratory, because it's very difficult to make such nanocrystals in different arrangements, like in chains. And there is a very special electron microscopy technique called electron holography, which can be used to measure and visualize the magnetic field inside these crystals, nanocrystals. So here is the magnetic induction map. So this is the product of this electron holography experiment, and here is the projected magnetization of this chain inside the bacterium. As you can see, all these lines run parallel to the length of these little crystals, and each crystal is a so-called magnetic single domain. So the bacterium is very efficient because it makes the crystals with the right sizes and puts them in chains. If these crystals were much bigger, then there will be different magnetic domains in it. So the orientation of the bacterium wouldn't be as perfect as is with this arrangement. Okay, so once we characterize these crystals, we want to know how the bacterium controls the growth of these crystals. And that's what biologists have done. This is not our work, but we just contributed to it by looking at these minerals, the crystals. Here is the bacterium, and now we know that first the bacterium makes a vesicle that is surrounded by this membrane, then pumps a lot of iron into this vesicle. Here is the iron, and starts to grow a crystal. And the crystal grows with a specific shape, and once it's large enough, it stops growing, and then it gets attached to this filament. So this is why it's in chain, because this green thing is a protein filament, and these blue proteins just clamp the crystal to the fiber. So actually all these marks here, these MEM-A, MEM-F, MEM-G, MEM-J, and so on, they are all specific proteins. So the bacterium makes certain proteins that control all these steps of the biomineral formation. So it's a complicated process, but this is a process that can be best studied in this single cell organism. You can imagine that studying how our bones form is a lot more complicated, and you cannot experiment with humans like you can with bacteria. So anyway, we know that which proteins play some parts in these different steps of the biomineral formation. Let me show just one example. Here is the wild type bacterium. That means that the original one, as we caught it from the sediment. And here is a mutant, a genetically engineered mutant, and only one gene is missing from it, this MEM-J gene, which is responsible for making the protein here. So what you see is that all these crystals have the same sizes, same shapes, even the number of crystals in a cell is about the same as here. The only difference is that they are not in line, they are not in chain. That's because this protein is missing from the bacterium, and this protein is like the curtain hanger clips that ties it to this fiber. So this is how we know what these proteins do. Of course, this is the most spectacular example, because the other examples are not as clear-cut, because all these genes and proteins, they have some synergic effects, but that's just on the side. So once we know how the bacterium makes the crystals, we want to use this information for making our own magnetic nanostructures in the lab. And that's what we did here. at this university, we collaborated with the molecular biology group, and who are very good in modifying the flagella of the bacteria. So here is the model organism, which is a salmonella, and as you see, it has many, many flagella. The flagellum is the organ of motility of the bacterium. And if you look at this flagellum in a cross section, you see that it's made of just one protein, this single protein called flagellin, and it has one part, this D3 domain. You see it's repeated all the way here, which is quite easily replaceable. So the idea that we had, the concept, was that we are going to replace this with something else, and that will be repeated all along this flagellum here, and we replace this with proteins that we knew from magnetotactic bacteria. We knew that they bind either magnetite crystals or they bind iron atoms. So that's what we did. We created some mutant bacteria. These have flagella that are already modified, either to tie magnetite nanocrystals or to bind iron atoms from solution and to make magnetite that way. So the end result would be a filament, a bacterial flagellum, decorated with magnetic nanocrystals. And actually it worked. So these are already mutant bacteria. I just put in this picture because I like it. It's nice that these are 4 bacterial cells, salmonella. They have lots and lots of flagellae, and all of these are already mutants. They are designed to bind magnetic nanocrystals. And this is when we add the magnetic nanocrystals. Here you might see these faint lines, which are the filaments, the modified filaments, the mutant filaments. And these bright features on them are just magnetite cubes. So we tied magnetite cubes to it, and we made such a kind of a cobweb of magnetite nanocrystals. So this is just an electron tomographic reconstruction. So it's like a spider's web, and some parts of the web are decorated with these magnetic nanocrystals. Well, actually, our intention was originally to cover these fibers totally with magnetic material and make magnetic nanotubes. It didn't work out that way completely, but still it's interesting. For example, if you measure the magnetic properties of this material, I'm not going to explain this in detail, but the main thing is that the green data are for magnetic nanoparticles and filaments separately, whereas the blue one is the sample that I just showed you, where we tied the magnetic nanoparticles to the filaments, and they behave very differently in magnetic fields. So when you have a liquid that is flowing and you create a magnetic field perpendicular to it, then there is a strong resistance to that. So we haven't used this material for anything yet, but it might find some practical application. Finally, I'm going to show you one more example that's a little bit different, but it also looks nice. So this was also made in the laboratory. These are, as you see, this is 50 nanometers. These cauliflower-looking things are also magnetite. It's called mesocrystals, which means that each of these component grains is in the same crystallographic orientation. They have specific sizes, and they are also in chains. So that was also made in the laboratory. So this is how we started in the environment. We collected some mud, isolated some bacteria from it, learned how they do their trade, how can they find the magnetic north, and what are their crystals like, characterize them, and then we use this information for making some interesting nanoscale structures, magnetic nanostructures. So I hope you like this little trailer of some of the work we are doing in the group. And if you are interested in any of these aspects, then you are welcome at our university. And finally, just one more slide. We have a web page where you can see all the research groups at our Faculty of Engineering. This is the, if you want to be a PhD student and you find information about our 2 doctoral schools at that address. And some of the more prominent research groups are also listed here with their web pages. It doesn't show the whole diversity of research at our faculty, it's just a small sampling, but since I don't know what is your field and what are your interests, I suggest you browse in the research group section of our web page and find the topic that is of interest for you. So thank you very much for your attention.